Hi, this is Miss Litton, and this is lunchtime, but a bunch of honors bio students that are amazing are here. Say hi. Hi. And we are reviewing the second half of chapter 27. So I made this little list so that you could kind of check it, make sure, like, do I know this? Do I know this? Do you know all of these little pieces of information, these sections? Remember, I gave you this presentation. It's in Google Stream, this Google Classroom, in the stream right now, if you would like to have that. So if you were going to look over that list, why don't you ping pong back and forth with your bio buddy right now and see what you can come up with on the fly about each one of these items and see if you can remember them. And I'll just pause for a minute to give you a chance to do that. Go ahead. All right, so I had a few minutes. Come back to me. I just wanted to make sure you're in your head so you knew, like, okay, what do you feel strong on? What do you feel like you need to give put a little more time into? So let's start with fossils. So we know fossils have to be 10,000 years old to be a fossil, and a fossil could be, any preservation of life in the past. It could be, a wormhole that's left in the soil. It could be an actual shell. It could be a cast of a shell. So we just want to remember it could be pollen, it could be insects, and let's try to remember what are some of the different types of fossils we actually saw. What's something you might see, let's say, an insect in? Amber, good. What's something you might see a woolly mammoth in? Ice, good. And then when we looked at fossils, do you remember when you have layers of rock? What is that called? Strata, or what kind of rock? Sedimentary rock. You might see things like where it's dry, but you still see seashells, so that tells you at some point, right, that area was covered with water. Good. All right, what are some other fossils? Did we see some other ones?
Petrified?
Yeah, could be petrified. Did we see anything else? Yes. Casts and molds, perfect. Okay, so you got that down. When we look at the fossil record over time, and so this kind of brings in the geologic time scale as well, it looks that in the oldest layers that the organisms are much more simple, and that you see a gradual transition in form over time. And when we look at the way things could possibly have evolved, you do see unicellular organisms before you see multicellular organisms. You see algae before you see plants that are on land. You see amphibians before you see reptiles, right? So you can see that sequencing, and then one really good example was the horse evolution, where you can see transition over time, and that probably matches where the continent was and the type of plants that they were living. Like when you look at flora and fauna, the flora, what kind of plants were available. So it was better to be larger than smaller. Okay, next thing is these transitional fossils. Now, do you remember the two speeds? This is something I introduced, and then we looked at it at the end as well. The pace of evolution. See if you and your bio buddy can remember those two paces.
Stable, equilibrium. It started with a P or a C.
Punctuated equilibrium. I think punctuated is very, very quick. And then gradualism model. Which one are you going to see the most transitional fossils? Gradualism. Yeah, when you have that slow change over time, you're going to see those transitional fossils. And there are currently transitional fossils between fish and amphibians, between amphibians and reptiles and mammals. In punctuated equilibrium, it's very rapid change. And by rapid, how rapid are we talking about? A million years, yeah. But in the geologic time scale, a million years is pretty quick. Okay, we also talked about when we date these fossils, there are two types. There's relative and absolute. Relative dating, right, you don't have an absolute number. You're just saying, well, this layer is old on the farther down, so it's older, and the layers on top are what? Newer, younger. You could use an index fossil to help you with that. Like if trilobites are only located during a different time, then you can say, oh, all the rock layers above this must be younger, all the rock layers before it are older. You can take continents and match up the layers and see if they match, and you can help infer dates, timing from that as well. Absolute dating, though, remember, that's when you're going to try to give it's not like a minute date, but an absolute date is in the millions of years when you would expect, and you would have a window of time. And that has to do with what? What are you doing in absolute dating? Carbon is an example, but it's half-lives and isotopes. Because like currently, let's say, what percentage of a substance should be a particular isotope, then the older it is, there's going to be less of that isotope because it's going to break down. So when we look at the ratio between two isotopes, you can tell how old it is, okay? And the time it takes for half of it to break down, that particular more unstable isotope, would be its half-life. Yes? Okay. So then we started talking about the geologic time scale. So right now, what era are we in right now?
Cenozoic.
And we're going to remember, because we're going to think about, we like to what? Drive cars. So that goes back how much? 65 million years ago. So when we talk about the Cenozoic era, we're saying it started 65 million years ago and it's still ongoing today. Okay? Before the Cenozoic era was the Mesozoic. It ended at 65 million years ago because that's when the Cenozoic started, but it started when? And how are we going to remember that? That's when school gets out. And before the Mesozoic era was the Paleozoic era. And we remember old and pale people like to eat dinner about when? Yes, okay? Then everything before that is about 90% of the Earth's time. And everything before that we're going to call what? Precambrian, okay? And in Precambrian, we're going to start when the Earth started, which is about 4.6 billion years ago. And about 1 billion years later, we had our first what? Cell. And from that first cell, was it an autotroph or a heterotroph? Probably a heterotroph. And then an autotroph developed later. Oxygen revolution about what? 2.3 billion years ago. And then another billion years later, we had the symbiotic hypothesis, and we had eukaryotic cells. By the time you're at the end of the Precambrian era, so you want to transition now into the Paleozoic era, plant-wise, what are we starting out with, plant-wise, in the Paleozoic era? Algae. Okay? Animal-wise, what are we starting out with? Invertebrates, right? So when you're here, right here at the starting point, plant-wise, we are at algae, and then animal-wise, we are at invertebrates. Good. By the time we get to the end of the Paleozoic era, plant-wise, we are now at early something. What are we at? Early, remember? Gymnosperms. Okay, and those are like Christmas trees and Hanukkah bushes, right? Okay, animal-wise, where are we at? Early what? No, not early. Early reptiles. So when you look, what happened during all of the Paleozoic era, you went from invertebrates to then being what? Vertebrates, and then probably what next? Fish, and after fish, amphibians, and after amphibians become these early what? Reptiles. Okay? So then the next era, you are starting with early gymnosperms, and you're starting with early reptiles, and by the end of the Mesozoic era, what did we get to? Okay, I'm at gymnosperms, so let's think that out. Nope. Angiosperms. Okay, so I'm thinking you need to review that a little bit because that was not coming to you. Okay, and reptiles were early what? Mammals. Okay, so that all happened during your Mesozoic era. So we went from gymnosperms to early angiosperms. Angiosperms are plants that have what? You know, flowers. So here's a gymnosperm I'm drawing for you right now. And then here is an angiosperm I'm drawing for you right now. See the difference? Okay. All right. Then you start with early angiosperms in the Cenozoic era. And what do we end with? Herbs. Here we go. And we start with mammals and we end with what? Humans. As if we're at the apex of everything, which we probably are not. Okay. Got that? Ping pong back and forth with your bio buddy right now about that. Make sure you understand that. Go ahead.
Okay. So algae was okay, great. Cambrian is 90% of the Earth's surface. So it started 6 billion years ago, and started with algae 42, and then that goes into the algae and algae
All right. So I really combined 2 pieces of evidence in this first one, fossils and the geologic time scale, looking at those 2 because they are kind of intertwined. The next one was biogeographical evidence. And what this is that certain organisms evolved wherever they had the right environment where it would work for them, right? And so at the time, we know that when mammals were evolving, the continents, were they together or apart? When mammals were evolving, where were they? Apart. They were separating. And this gives you an example of, or evidence for evolution, because that explains why there's so many unique species of mammals on so many different continents, but maybe there's a universalness of a type of fern. Because when ferns were evolving, all the continents were together. And so it makes sense that you would have a lot of similar fern species on the different continents. Okay, so that is your third piece of evidence. Your fourth one is all about anatomical evidence. And there are 3 types of anatomical evidence. Homologous structures. Homo means what? Same. So homologous structures are the anatomy is the same, like humerus, radius, ulna, carpals, metacarpals, phalanges in the forearm. And since they all have similar bone structure, they may have longer or shorter phalanges or fused phalanges. That must mean they come from a common what? Ancestor. Do they have to do the same thing with their forearm? No. Could they do the same thing with their forearm? Sure. Bats and birds do the same thing with their forearms. So not only are they homologous, they are additionally what? Analogous. But analogous is not a piece of evidence for evolution. Analogous just means I go through life the same way you go through life. I swim through it. I fly through it. I walk through it. Insects have legs and we have legs. But do we have homologous structures for legs? No. Totally different. Okay? So therefore, we do not have a recent common ancestor. All right? So that is evidence 1 is homologous structures. So do not use analogous on any test, right? Because that's not a piece of evidence. Right? The second part of anatomical structures were vestigial structures. So these point, the reason why it's evidence is it shows we could have used something in our ancestral past, but as it did not have the same importance as we evolved, then we kind of lost it. So for instance, we have a tailbone, which means in our ancestral past we could have had a what? Tail. Okay? So when you look at an ostrich is a bird and it has what? Wings, but does it use its wings for flying? No. So it's like they're vestigial, they're greatly reduced. I always give the example about salamanders, because salamanders have vestigial eyes, but they're not working eyes. The particular salamander, not all salamanders, but salamanders in caves. So that tells you at one point they lived somewhere where they needed eyes. Now they're in the dark cave, they don't. I saw your hand up, Michaela. Yes?
So it means like they needed this structure before, but they don't need it anymore.
Right. Like some people have the muscles to move their ears, right? So they can control them and move their ears, and some people like me cannot. So if they have, some people have the remnants of that ability to control how their ears move. Okay, so it points to your ancestral past. Those are vestigial structures. Okay, and the third piece of anatomical evidence is embryological development. One, you can kind of see evolution on fast forward, as you know, you have gills at one point, as you have a tail at one point, but also you're just comparing the embryonic structures themselves. The more similar you are in embryological development, the more likely you have a common ancestor. Okay, so we looked at fossils. What was the next thing we looked at? Before biogeographical, we also, no, before that. Geologic time scale. So fossils combined with geologic time scale. Then we looked at biogeographical and now anatomical. And remember, anatomical has 3 rungs to it, right? Homologous structures, vestigial structures, and embryological. Okay? Then the fourth one, and the one that's most in use today, would be biochemical evidence. And if you have access to it, right? Because a fossil of a shell, you may not have any of their DNA, right? And you're going to have to just use structures. But biochemical evidence is comparing DNA sequence, you're comparing proteins and looking at the amino acid sequence. And from that, you would assume the more similar our DNA or proteins to another organism, the more closely related. And you can use that to build cladograms, for instance. This is a structural cladogram. Like, we talked about being in a club, right? So everybody but the hagfish, because they're jawless, everybody else gets to stay in here because everybody has jaws. From here up, everybody has jaws and lungs, except we dropped off the fish. From here up, everybody has jaws, lungs, and claws, except we dropped off the salamander. Okay? So by looking and organizing it that way, it can give you the phylogeny, the history of who came from who. Okay? And you can also do that with protein sequences or DNA sequences as well. All right? It's funny, I have a couple repeats in here, and I don't know why they're in here, but they're in here. Okay, the next thing, interesting. The next thing, weirdness. I'm going to go with it, though. Weird. This was supposed to be here. Okay, so evolution is a result of 4 factors. The potential to increase in your numbers, variations in your DNA, competition for resources, best adapted one wins. So we started talking about microevolution versus macroevolution. So why don't you talk about that with your bio buddy? What's the difference between micro and macroevolution?
Micro is like
Weird.
like if all of the species kind of micro is like tiny but then it's micro, but then if it happens on more like a widespread, then it's macro.
So weird. I have these random repeats. Okay, so microevolution was like the peppered moth. They're still the same species. It's just, do you have more black ones or do you have more white ones? And we saw what was putting pressure on whether the black one or the white one was more numerous had to do with their environment, right? So when there was no pollution, it was better to be what color? White. But as the pollution started coating the tree, then you saw the black allele go up and the white allele go down. Yes?
If the black and white moths would no longer reproduce with each other, would that be micro or macro?
If they no longer, if white stayed with white and black stayed with black, and so they were just reproducing in and amongst themselves, then that would be macroevolution. Good. Okay? And then when there were better controls on pollution, then it became more popular, right? The white ones were surviving because other organisms weren't seeing them to eat them. Okay? And so that is microevolution. Macroevolution is if you change to a totally new species, and we'll talk about that at the end. That was speciation and the different ways and factors that can contribute to speciation. But our next thing we talked about was about Hardy-Weinberg, okay? And we talked about looking at allele frequency. So it's like how many dominant alleles are in your gene pool, how many recessive alleles are in your gene pool. And instead of using like big B, little b, then we started just, let's use the word P's and Q. So P and Q, okay, when you just look at this equation, okay, then that's in your gene pool. That's if each individual took their 2 copies and threw them in the pool, okay? Mix around like a big pot, and then you reach in and grab 2, that's going to be a person, right? And that person could either be homozygous dominant. If I were going to use eye color for that, I would say that that would be what? Big B what? Big B. Or they could be heterozygotes, which would be what? Big B, little b. Why do I have a 2 right there? Yeah, you could have it be big B, little b, or little b what? Big B, which is the same thing. That's why it's 2PQ. Okay? And then homozygous recessive would be like blue eyes, little b, little b. So this is a trait right here. So this is the resources you could get a trait from is your gene pool. So you look at not individual crosses anymore, Billy and Betty making a child. You're looking at the whole population reproducing with each other. And so if everybody is like potential alleles, whatever genes they have, whatever they're carrying, throw them all in the gene pool and then see what you pull out. You can predict, well, based on this allele frequency, I bet this percentage of the population will have the dominant trait, this one will have the recessive trait. Yes?
In like brown eyes, is there a way to tell like, for sure it's homo or heterozygous? Or is there no way to
No. I mean, you could tell for sure, like if you're a brown-eyed person, you have to look, we're going to be looking at pedigrees when we talk about it with genetics. And so if one of your parents has blue eyes, then we know you're a heterozygote. Because that's all they have to give is the little b. So we'll be talking about that when we get into our genetics unit. Okay, so when you have a problem, okay, here's a practice problem for you. The right-handed trait is dominant over the left-handed trait. If that is true, in a classroom, let's say we count everybody up and 83% are right-handed. So that means 0.83 are right-handed. So then answer these 3 questions for me based on that information, and I'm going to pause it so you can get a chance to think it through. Try to do it by your lonesome, all by your lonesome. And then once you have it, then talk to somebody else and you can compare your answers. And I'm going to pause just for a minute. So the way I would approach this problem is I would say, okay, 83% are right-handed people. That's true. But I don't know if they are right, or they could be what? Right, left. So the only thing I can work with are my lefts. So how I would approach that is I would say, okay, so there's 100% are all the students. Right? And how many of them are right-handed? 83, right? 83%, or sorry, 83%. So 100% minus 83% leaves what? 17%, or that is equal to 0.17. Do you agree with that? Okay, that 0.17 is equal to my left-handed people, and in my equation, my left-handed people are the Q squareds. They have the 2 recessive alleles, right? So now I know Q squared equals 0.17, so I'm just going to take the square root of that, and I'm going to solve for Q. So did you take the square root of 0.17, and what did you get? 0.41? 0.41? Yeah, 0.41. Well, I know if I'm not a Q, I'm a P. So what do the P's have to be since the Q's are 41% of the alleles? They have to be what? 59. Because that equals 100, right? That equals 1. This is what's in my gene pool. This is my pool. Coming out of that, I could have P squareds, I could have 2 PQs, and I could have Q squareds. This, I know, is 83% of the population, and this, I know, is 17% of the population, right? So I just solved for P and Q, and my question was, my third question is, how many are heterozygotes for it? So that's when I need to go, okay, 2 times 0.59 times 0.41. And what does that equal to? 48%. Point what? 48. Okay, so 48% then are heterozygotes. Does anybody have a question about that you want to ask me? Yes.
So can you go back to the third question?
Yes. It's right here. Yes.
Oh, when it asks for frequencies, you want the decimal or the percent?
Probably the decimal. Yeah, that would be better. Yes.
So in these type of problems, you're dealing with the whole dominant trait, but if it were to say allele, that would be like just
the gene pool. Exactly. I'm just trying to give you the hardest one so you can do all the other ones if you can do the hardest one. Yes.
Can you go back to the side with the calculation?
Yes, I can. Okay? Now, Hardy-Weinberg law says if you can freeze allele frequencies and they don't change, then you can stop evolution. If you can't freeze the allele frequencies, they will change. So, 5 fingers, what causes evolution? 5 fingers. Small populations, right? So if you want to stop evolution, it has to be a what? Large population. Because small populations are susceptible to genetic drift, founder's effect, and what was your other one? Bottleneck. What was the next one? What causes evolution? Non-random mating. So to stop it, it needs to be what? Random. What's your next one? Mutations. So this one needs to be no mutations. What's this one? Gene flow will cause it. So this one needs to be no gene flow. And what's the last one? Selection. So this one needs to be no selection. Okay, and then since you can't do that, evolution occurs. Last thing I want to remind you is that we're changing things very rapidly, so they can't get caught up, right, and causing extinctions. Allopatric, sympatric speciation. What's the most common? Yeah, allopatric, you have some biogeographical divide, like a river. Here, it's mostly about what? Chromosomes, good. And then we talked about adaptive radiation and going into all the different niches. And then that was it, because we already did the last part. Okay? You're smart. Awesome. I will see you mañana. Make good choices. We missed you if you weren't here.